During flawed cellular division, cancer cells frequently missegregate their chromosomes. This mechanical error leaves behind fragmented pieces of DNA, isolated in the cytoplasm as small structures known as micronuclei. These micronuclei are highly unstable. When their fragile envelopes rupture, they spill raw, double-stranded DNA directly into the cellular cytosol, instantly tripping the cell's internal pathogen sensors. The canonical response to cytoplasmic DNA is the activation of the cGAS-STING signaling pathway. The STING protein acts as an alarm, triggering a rapid release of type 1 interferon into the surrounding environment. Biologically, this pathway operates as an immune beacon. By releasing interferon, the cancer cell flags itself as a threat, recruiting T cells for rapid, targeted destruction. If chromosomal instability continuously triggers this alarm, highly mutated tumors should be rapidly eradicated by the host immune system. The clinical data shows the exact opposite. Tumors with the highest rates of chromosomal instability are the most aggressively metastatic. The survival and spread of these highly unstable tumors exposes a structural flaw in our prevailing assumptions. The activation of the cGAS-STING pathway does not guarantee immune clearance. A recent study investigated exactly how aggressive cancer cells survive this continuous cGAS activation without being eliminated. The cells do not simply mutate the STING pathway out of existence, they adapt to the persistent alarm. They execute a definitive intracellular signaling rewiring. The tumor cell permanently shifts its downstream response away from the anti-tumor interferon pathway, redirecting that signaling energy toward endoplasmic reticulum, or ER, stress pathways. The study investigates the concept of non-cell autonomous metastasis. This model examines how chromosomal instability allows cancer cells to manipulate the behavior of the surrounding environment, shifting the focus away from the internal traits of the cancer cell itself. Persistent chromosomal instability hijacks host biology. It forces the STING pathway to abandon its role as a suicidal alarm system and operate instead as a pro-metastatic engine. To test this mechanism, researchers utilized 4 distinct syngeneic mouse models, spanning breast cancer, colorectal adenocarcinoma, and melanoma. In immunocompetent mice, tumors with high chromosomal instability produce substantially more metastases, but in severely immunocompromised mice, this metastatic advantage completely collapses. Targeted genetic knockouts isolate the specific pathway. When the cGAS gene is deleted from tumor cells, metastatic rates plummet. Deleting the STING1 gene from the tumor cells yields the exact same reduction in metastatic behavior. Artificially restoring STING expression in these models produces a strict dose-dependent return of aggressive metastases. Ultimately, the metastatic spread of these highly unstable genomes depends entirely on the active involvement of the host immune system. The critical variable is the timeline of the signal. A standard pathogen triggers a brief, acute STING activation. Continuous chromosomal instability forces chronic, unyielding activation. To survive chronic STING signaling, the cell develops IFN tachyphylaxis, drastically downregulating the anti-tumor interferon program. Signaling energy is forcefully rerouted, hyperactivating the unfolded protein response and ER stress sensors. Knocking out these new routes, like IRE1-alpha or PERK, severely stunts metastases. Knocking out ATF6 completes the evidence. The cellular ER stress machinery is functionally critical for the cancer to spread. Crucially, disabling these ER stress sensors reduces metastasis without impacting the baseline cellular proliferation of the primary tumor. Tumor cells weaponize the chronic STING signal. They construct an entirely new, ER stress-driven survival pathway to maintain their viability. The ultimate output of this ER stress rewiring is a highly altered secretome. The tumor begins releasing specific immune-modulating factors like CCL2, CXCL1, IL-11, and ApoE into the surrounding tissue. To prove these secreted factors actively communicate with neighboring immune cells, the research team developed a computational method called contact tracing. Contact tracing uses single-cell RNA sequencing to match a tumor cell's ligand expression to downstream transcriptional responses. At baseline, stable tumors attract inflammatory cells, maintaining a hostile environment. But for unstable tumors, the mapping shifts, revealing a total reversal that recruits highly immunosuppressive suppressor cells. This environment forces the accumulation of exhausted, dysfunctional T cells, effectively rendering the host immune system blind to the tumor's expansion. The stress secretome engineers an immunosuppressive shield around the cancer, fulfilling the non-cell autonomous requirement necessary for systemic metastasis. We can map this precise biological sequence from engineered murine models directly onto human patients, utilizing spatial transcriptomics and single-cell analysis of triple-negative breast cancer. The study proposes a targeted biomarker classification, isolating tumors that are CJAS low and STING high versus those that are CJAS high and STING low. Human tumors exhibiting high chromosomal instability and high CJAS activity, combined with low tumor cell STING, perfectly mirror the murine stress signatures. This specific CJAS high, STING low signaling state is strictly correlated with aggressive, heavily immunosuppressed tumor microenvironments. This Kaplan-Meier survival curve highlights the ultimate clinical consequence. Patients carrying the CJAS high and STING low tumor classification experience a steep drop in distant metastasis-free survival. The signaling tachyphylaxis and pathway rewiring observed in mouse models drives lethal metastases in human patients. These mechanics challenge a widespread assumption in clinical oncology that administering STING agonists is a universally optimal strategy for treating cancer. For early-stage or STING-naive tumors, activating the acute interferon pathway with an agonist remains a viable method to trigger immune clearance. But the data reveals a therapeutic inversion. For tumors with tachyphylaxis, administering STING agonists is futile. It actively accelerates the unfolded protein response and promotes metastases. The research points instead toward utilizing STING inhibitors to silence the chronic alarm signal at its source. This opens a parallel strategy, combining stress blockers with immunotherapy to dismantle the immunosuppressive secretome. The causal chain is clear. Chromosomal instability creates micronuclei, generating chronic STING activation. This forces interferon tachyphylaxis, redirecting the cell to the unfolded protein response, which engineers an immunosuppressive microenvironment, ensuring metastasis. Mastering the transition from cell-autonomous genomic instability to non-cell autonomous immune subversion is the mandatory next step for precision oncology.